And thanks also to Alistair for really introducing engineering at the DNA level. What I want to do now is to shift the focus slightly to think about cells. And to do this, I want to introduce this time-lapse video. It's from the BBC Natural History Unit. You may recognize it from various sort of David Attenborough films. But you'll see here that you've got this leaf-like structure with this stem growing from the end of the leaf. And as it grows, you can see the small nub of tissue at the bottom here. And that will continue to grow and expand to make what becomes recognizable as a pitcher plant. So as it continues to grow, you've got this coordinate activity of all these cells, millions of cells, all coordinately dividing and expanding and differentiating to make what is effectively a coffee cup structure, in the sense that you've got this lid, if you like, a cap on top of a hollow structure. And this process, as it grows, creates this hollow structure, opens up, and then, of course, it fills with water, and its biological purpose of creating a sac of digestive material that traps insects and feeds phosphorus to the plants becomes clear. But this effectively is a coffee cup. And when you think of a coffee cup, there's something like 200 billion coffee cups made each year on the planet. And as humans make these structures, they do it in a very different way to plants, of course. You have a process whereby you've got the mining of materials, the collection of natural resources, the creation of templates, and the automated assembly of these elements in high throughput. And you're making something which is actually quite similar to the biological equivalent. You have in the coffee cup a curved lip, just as you have in the pitcher plant. In the coffee cup, you have these low-density polyethylene linings to stop leakage. Similarly, you have wax epicuticular material deposited on the inside of the pitcher plant to line the insides to make it waterproof. It's actually a very similar structure, but couldn't be made in more different ways. The design and assembly of the pitcher plant is a collective, a social process where you've got the individual cells that make up that, millions of cells that make up that pitcher plant, start off from a small group, maybe a single cell, which then undergo this sequential process of division to create daughter cells, where the division of cells in this cartoon representation involves duplication of the DNA, physical segregation of the DNA, deposition of all the materials that are required to create two daughter cells. And that process that Alistair also referred to, this proliferation, is something which is self-organizing and pre-programmed. And you see the generation, the splitting, the bifurcation of information systems and the propagation of those physical systems through growth. And as it continues to grow, you've got the reprogramming of those genomes. And again, as Alistair pointed out, each cell has an identical genome, but it's in a different state. So as the cells divide themselves, as they proliferate, they adopt different fates. And we know from many years of developmental biology in plants that the individual cells, say for example in a pitcher plant, undergo interactions with their neighbors to adopt a particular fate. And if this schematic cell, for example, is shown to divide, the two daughter cells are shown here. And these two daughter cells are, by definition, in different contexts. They're associated with different cells, and you get this bootstrapping effect where you've got differentiation or different states of gene expression emerging by this process. And in fact, the DNA code that encodes that shape is the result of a social process. So if you think of that coffee cup and think of its context, it's more the interaction between the elements in the system that drive the differentiation process. And we're thinking of a Starbucks coffee cup as an example, and I'm stretching the analogy here a bit, but if you think of that, the organizational principles that underpin the creation of a biological version of that is more similar to what happened with the growth of the Starbucks network. The Starbucks collection of shops and the whole industry around Starbucks is based on individual transactions. Individuals making choices, exchanging cash for resources, and the propagation of that social and economic network drives the formation of Starbucks from a single shop in the 70s to this sort of dominating coffee culture across the planet. And in a way, that's what's happening in the biological coffee cup. In the pitcher plant, you've got individual cells proliferating, interacting, differentiating to describe its own program. So it's drawn from within by this highly social process. And of course, it gives rise to all kinds of different variations. So the DNA that it encodes those interactions can encode very different endpoints, very different structures. Here we've got a whole range of things which are closely related, but give rise to different outputs. And the process of reprogramming that is now a major challenge for biology. And in order to address that challenge, biologists are increasingly turned to formal engineering. And I'm going to talk about plants as an example, but the same thing really applies to animal systems in different contexts. But of course, in plants, crop plants, for example, we know there's a whole series of issues. We've got very slow generation times, generally speaking, compared to, say, bacteria. They're complex genomes. There's lots of redundancy. They're quite difficult to work with in the laboratory. They're complex, and they're hard to actually visualize directly under what we use in modern biology is quantitative microscopy. So they're difficult to use, but modern plants derive from ancient antecedents. So if the planet was 500 million years older than it is today, and you looked outside, this is what plants would look like. And in fact, if you look at the entrance to the Robinson College, in the cracks in the pavement, particularly against the walls where it's a little bit damp, you see relics of these ancient plants. And I'll show pictures of them in a minute, in fact. So this is the sort of form that plants would have taken about 500 million years ago. Sort of flattened structure, no roots, no shoots, no flowers, no seeds, but they have these sexual reproduction systems, ball-based systems at the top of a flattened structure. So they basically drag themselves out of fresh water onto land, and they are dealing with the challenges of a terrestrial environment, but have a very simple genome structure, very simple architecture. And so we've exploited that in the lab. This is the kind of plant that you'll see in the front of Robinson College. It's a weed, obviously. It's something that we use increasingly as a model plant, as a simple form of something to manipulate for engineering purposes. And I'll talk a little bit more about what all this means. So if you look at the top surface of the plant, it has this very simple reproducing structure. So you've got these little chambers, and these donut-like regions correspond to air chambers. So if you look at that top surface of the plant, each one of those is a modular structure, a bit like the equivalent of a leaf in a lower plant. And so each one of these structures involves this chamber-like architecture. The donut-like structure is this air pore on the top. There's gas exchange and photosynthetic cells in the middle here. And so that's the equivalent of what would be a leaf in a higher plant. You have no roots, but these individual hairs at the bottom part of the plant. You've got the ability to propagate not by making flowers and seeds, but rather by making these features which erupt from the surface of the plant and grow into these sexual features. And they make male and female parts. They're haploid. They cross and create spore capsules. So each one of these little yellow structures here is a package of spores which contain these single cells, which look like this. So there's something like 250,000 of these single cells inside those spore capsules. And when we think of seeds, this is the equivalent in lower plants, where they'll cross and they make millions of progeny. And that's how they manage to get around, of course. They propagate. And each one of these little cells is a package of DNA and resources which allow it to grow. So if you put it onto a sterile media, it grows into a cell, which looks like a recognizable plant cell. We now have green chloroplasts. Within 2 days, it's gone from this, which is one of the ungerminated spores, to this, the first cell division. And that division process keeps going. And just as you watch it, it will divide under the microscope and create this larger scale structure. And after a few days, it ends up as this one of these little structures here. And this is on an agar plate. So it's a bit like those of you might be familiar with yeast or bacteria, growing them on agar plates. This is doing the same thing in a plant context, but making a baby organism rather than a colony. And so it's very useful and accessible for experiments. And the thing that's driving that whole process is the genome. And as Alistair referred to the human genome, here we have the genome of one of these primitive plants. It's much smaller. It's 280 million base pairs instead of 3,000. There's about 20,000 genes, so it's actually not that much simpler, but it has a more redundant genome with genes involved in the cell-to-cell arrangements and how it defines its morphology. So it's a relatively simple and accessible system. And what the field is now doing is, with systems like this, is to adopt more systematic ways of trying to rejig, resynthesize, and reform the whole genome in a way that you can reprogram growth, development, and metabolism. And so we've actually taken some inspiration, or see parallels at least, with what happened in early microelectronics. And this is the first integrated circuit from Jack Kilby and what became Texas Instruments in '58. And so in these early days, these are obviously crude, bespoke devices, but within a few years, by the early '60s, you had planar transistor technology. The first simple integrated circuits were made. You had the adoption of both electrical and physical standards that were used to encapsulate and modularize those components for circuit assembly. You saw the development of both technologies for software design and automated layout of circuits as these circuits became larger in scale. And at the top here, we've got this first planar transistor right through 60s, 70s, 80s, 90s, and 2000s, with the increasing scale of integrated circuits and the number of logic elements involved. And it's on a similar kind of trajectory to the kind of increase in scale that we can work with DNA now. And so modern circuits, of course, have billions of elements in them. And it's beyond the kind of an individual to deal with that. You need these kind of automated skills. And that's essentially what's happening in biology. People are starting to look at how you can use modularity and engineering principles to deal with the kind of scale of complexity. And if you think about biology, you've got quite a different set of issues here. So in the case of a circuit, for example, you've got a layout which is dictated or constrained by insulators and conductors. In the biological context, and this is a diagram of protein interactions inside a yeast cell. So this is the interactome inside a yeast cell with the different proteins. They're all in the same sac. So they all can have the potential, if made, to interact with each other. And what is the determinant here is molecular specificity. So it's not insulation as such, but molecular specificity. And the cellularity provides the insulation. So you've got different layers of complexity that need to be juggled in this case. And so in terms of trying to make the transition between the natural genomes, the evolved information systems which create these organisms, to a more engineering context, this idea of splitting DNA into different hierarchies, so taking natural systems, defining particular functions which can be put together to make logic devices or various types or functional devices, which can then be pieced together in circuits and systems and then ultimately implemented in multicellular systems, is something which is part of the synthetic biology paradigm, if you like, where people are adopting engineering principles or attempting to in a biological context, using DNA as the code effectively to drive these processes. And we have the benefits of abstraction and decoupling of fabrication and design, which come with this approach. So at one level, it's sort of a trivial mechanical aspect, but on the other, it's quite a revolution for a field that's been dominated by analysis of natural systems to move towards a more consciously engineering approach. And so this is an example from the same lower plant as I've given as an example. So one can take, for example, the genome described by sequencing principles, map some of the functions on that genome, and then create databases which describe the genome not in terms of its physical context, but in terms of its functional components. And use this as a way of creating a resource for automatically extracting parts and being able to collate characterization information. So it's essentially a database not for natural scientists, but a database for engineers. And using this kind of abstraction, you can couple that with, for example, standards for describing these functions, not dissimilar to what happened in the early circuit or integrated circuit design field, where you can start to create design systems, and you can see here these 4 base pair elements which define the ends of, and at the top here there's this colored schematic of an arbitrary and abstracted gene, where you can define individual pieces of genes, functional pieces, in a way that allows you to start pulling them together and assembling them in a particular way. So you can take these arbitrarily defined functions as DNA pieces and use new highly efficient techniques for putting DNAs together. And this is one particular example. There's a loop-like arrangement for piecing together individual parts to make gene circuits. And that's shown, the extension of this is shown on the next slide, where you can use this loop-like arrangement to start out with individual parts, put them into individual genes, string the genes together to make circuits at different scales by doing this iterative process of assembly. And I won't go into the technical details, but this is another step forward in the kind of things that Alistair was talking about. We're speeding up the work and making it independent of individual decision making. The assembly process is baked into the description of the parts in this way. And so the idea, or the aim at least, is to put together ways of working in biology which provide this sort of, might call it an equivalent of middleware, if you like, where we've got public research in natural systems and applications often driven by the commercial world, but in this case glued together by standardized systems and ways of working and open technologies, where obviously many electronic systems are bound together by open systems which bind together sort of basic research, which I've crudely called academia, which of course is beyond that, and gluing that together to electronics or applications at the other end. And in biotechnology, it seems increasingly important to adapt more accessible open technologies for this, because of the implications for these new technologies for sustainable technologies in the third world, for example. So we've, in Cambridge, have been adopting a consciously open approach. In collaboration with colleagues in Stanford, we've developed this legal framework for sharing these individual elements through open MTAs. And again, I can address that in more detail if people want to know more about it. But what I just want to finish off with is this idea that one can start to use or develop parts-based systems, integrate them back into biological systems in a way that starts to iterate this idea of having a design, build, test cycle in a biological context, where most of the biological systems that we're working with require some kind of software underpinning, that we can use these standardized parts to build DNA circuits of increasing complexity, but in order to test these, they need to be integrated back into a biological system and tested in some way. So there's a lot of focus on how you visualize the role of one set of genes in the context of a whole genome in a large-scale multicellular system. And this all comes down to imaging. And I referred to this briefly before, but it's increasingly important. And that's one of the reasons why these simple plant systems are so important. And in the 90s, there was this discovery that many living systems use fluorescence in a natural world to either to signal or adapt light. And there are proteins which are responsible for these fluorescent signals, which can be used as synthetic genes and used to color or decorate living processes. And here we've got tubes of protein down here, or bacteria which are expressing fluorescent proteins showing in a laboratory context. You can use these proteins, deliver them as DNA pieces into, in this case, plant systems, and use them to paint or decorate living processes in living organisms. And these are shown here, where we've got essentially a paint box of elements. Here, there's two sets of targeting systems used to label membranes or the nucleus. And you can see there's different colors in these different arrangements. You can use these systems to decorate. In this case, it's a portion of a plant where we've got the stem portion of a young plant, the young growing point or meristem, and the first leaf-like structures in the young seedling. And each one of these cells, you can see, is decorated by this yellow color, because we're expressing two different fluorescent proteins, a yellow and a green one. And it allows you to then get ratiometric values for each cell. You can work out what the quantitative expression or activity of a gene is in an individual cell, obviously with a field where you can capture thousands of cells at once. So it opens up the door to new ways of observing and quantifying behavior inside biological systems. And back to our simple plant, one of the advantages of this simple plant called Marcantia is that it makes these spontaneous clonal propagules. So in these cup-like structures, you have a bottom well where there's an embryo-like process that takes place which produces these small propagules. These are such a pernicious weed. If you go to a poorly maintained plant nursery, you'll see these everywhere. Once you start watering, these are called splash cups. So if you're watering the plant or there's rain, these small propagules splash out and propagate themselves. So these are essentially small nicely defined little structures which can be harvested directly from these plants and then produced in large quantities. And this shows one of these small propagules. You can see there's individual cells here. Each one of these sort of circles here is a single cell. You can label the individual cells inside the plants. You can generate, with different promoters from one of these databases, generate a color-coded model for what's happening here. And these, of course, are alive. So if you run one of these under a microscope, you see the growth of these organisms where you can generate quantitative information directly from the material as it's growing. So you've got a simple architecture, the growth processes are continuing, and you can interrogate it. And so here's another one, it's a bit older, the same one, a little bit older. So you can see the division and differentiation processes directly. And so to summarize, and representing what's happening in the field, there's this growing attempt to try and capture a more standardized view of biological systems, to essentially fit them into ways of working that are more directly accessible, and also accessible to this design, build, test way of viewing things. So clearly the field is starting to move towards a much more engineering-orientated approach to dealing with biological systems and thinking of it from the point of view of components, systems, the feedback relationships that are involved in maintaining larger scale systems, and trying to address this very difficult challenge of how do you take a code, a DNA code, and program a social system in this kind of context, where we have the benefit of molecular specificity and the ability to make things atomically precise via DNA code, but there's this issue of going through different layers of organization, if you like, to get to the end point, which involves DNA, cells interacting, and physical aspects feeding back on the way DNA is behaving as well. And so this is leading, I think, in the future to this idea that we can democratize some of these challenges, if you like, and this is something that represents, again, work that's going on in Cambridge at the moment, looking at spin-offs from this kind of approach and technology, where, for example, we've got bacterial cell-free extracts, which are now accessible, which don't contain cells anymore. They're essentially cytoplasm that's been extracted from bacteria and can be distributed freeze-dried. So it's the kind of thing that you can use in an electronics lab or a physics lab or an engineering lab. It's not stuck in a biological lab. There are all kinds of challenges that we're developing inside Cambridge, funding opportunities and challenges around instrumentation, trying to bring physical scientists and engineering individuals together with biologists, and looking also at the application of these relatively cheap systems, which don't require large resources, in Latin America and Africa, and building open curriculum materials for developing tools in that area. So I'll just leave it there, trying to outline the challenges of trying to deal with whole organisms, but also highlighting some of the spin-offs, and give credit to a lot of the people who've been involved with the work. And thank you very much.
Thank you.